Hello everyone, welcome back again to the series of solved previous year's ICAR PG examination for plant science, where we will discuss the questions on genetics and plant breeding. So this time we'll discuss the question paper of 2017. Let's get started. Hello everyone, welcome again to the series of solved previous year's ICAR PG examination, plant science, where we will discuss the questions specifically on genetics and plant breeding. Today we will discuss the question paper of 2016. So let's get started. Question number 1. The character not studied by Mendel is? So Mendel did not study the cotyledon color. Question number 2. The interaction between two alleles of the same gene is? It is known as dominance. If we talk about epistasis, it is the interaction between different genes of interspecific hybrids. Question number 3. Brassica juncea is a cross between, so it is a cross between Brassica nigra and Brassica campestris. Here we can see a huge triangle is there, which shows that Brassica juncea is an amphidiploid having chromosome number 18, a cross between Brassica nigra and Brassica repa. Repa is also known as campestris. Also we can see that Brassica napus is a cross between Brassica campestris and Brassica oleracea, whereas Brassica carinata, it is a cross between Brassica nigra and Brassica oleracea. Question number 4. Isozyme is? Isozyme is a codominant marker which is suitable for estimating the population genetics parameters and is also used for gene mapping. Next question. RNA is not involved in. So RNA is not involved in chromosome because chromosome is made up of protein and DNA. Question number 6. Cry gene is obtained from. So it is obtained from bacteria, Bacillus thuringiensis. These bacteria, they form crystals during their growth phase. From there, the cry protein is generated. Question number 7. C2 cycle takes place in. So it occurs in chloroplast, peroxisome and mitochondria at several steps. This implies the answer is all of the above. This C2 cycle is a light-dependent cycle, which is also known as carbon cycle or photorespiration. Question number 8. Hormone involved in cell cycle is? So it is cytokinin, which promotes cytokinesis or the cell division. Question number 9. Under water stress condition, which amino acid gets accumulated? So the amino acid which gets accumulated is called proline. Proline is able to maintain the protein integrity and enhance the enzymal activity. Question number 10. Test cross in supplementary epistasis is? So its correct answer is option number B, 1:1:2. We know in dihybrid cross, the ratio is 9:3:3:1. In case of supplementary epistasis, the gene interaction takes place and the homozygous recessive alleles, they are suppressing the both alleles. So this becomes 9:3:4. We know that these two genes combine, so the test cross becomes 1:1:2, taking both these genes together. Question number 11. Albinism in plants is controlled by? So it is controlled by a recessive lethal. First division of meiosis is? It is called reductional division. As we see here, 2n becomes n. Question number 13. When two homozygous parents are crossed, no genetic variation is found in? So the variation is not found in F1 generation. Question number 14. Initiation codon is? It is AUG, that is methionine. Question number 15. Embryo is? So embryo ploidy is 2n because it is formed without the process of meiosis and as a result of double fertilization. Aleurone layer is 3N, which is a protein-rich layer. Question number 17. How many mitotic divisions should occur in a cell to give rise to 256 cells? We know that 2 raised to power n divisions take place in mitosis. So if we require 256 cells, the cell should divide 8 times. That is, the correct answer is option C. It should divide 8 times to give rise to 256 cells. Question number 18. Linkage group of rice is? So the correct answer is option A, 12. We know that 2n is equals to 24 in case of rice. Hence the haploid set has 12 chromosomes. This implies there are 12 pairs of homologous chromosomes and 12 different linkage groups. Question number 19. Formation of embryo from synergid or egg cells is called? It is called apogamy. Question number 20. Chromosomal number found in a gamete is? So it is N. Closely linked genes that are functionally similar and structurally different are? They are known as isoalleles. If we talk about pseudoalleles, there are 2 genes with similar functions located very close on a chromosome so that they are genetically linked. In case of multiple alleles, they are formed of genes which are situated at same locus of a homologous chromosome. Question number 22. Cytoplasmic inheritance is due to the DNA present in, so it occurs due to the organelle DNA, that is the DNA present in case of mitochondria and chloroplast. So the correct answer is option D, both B and C. Question number 23. Which of the following is a GURT, that is genetic use of restriction technology? So the correct answer is option C, terminator technology, in which the genetically engineered plants, they produce sterile seeds at the time of harvest. Question number 24. Antisense RNA technology is used for, so it is used for gene inhibition. It prevents the RNA translation into proteins. Question number 25. Genetic engineering is associated with, so it is associated with plasmid. Plasmid is a small circular DNA molecule which replicates independently and forms a recombinant DNA. Question number 26. Presence of R line in A line is called, so it is a pollen shedder. Question number 27. Chromosome number of common bread wheat Triticum aestivum is? So the correct answer is option C, that is 2n is equals to 6x, which is equal to 42. A hexaploid Triticum aestivum has 42 number of chromosomes. Question number 28. Enzymes used in polymerase chain reaction is? So the correct answer is option A, Taq polymerase. It is a heat-stable enzyme which is extracted from a thermophilic bacteria called Thermus aquaticus. This enzyme is used in the step of extension, which is performed at a very high temperature. Question number 29. Heterosis is fixed in, so it is fixed in apomixis. Otherwise, these methods, they exploit the heterosis. Question number 30. The term Mendelian population is used for, so it is used for random mating population where the mating occurs randomly. Question number 31. RNA polymerase binds to, it binds to a promoter gene. Next question. An individual with an extra pair of chromosome in addition to its normal somatic number is called, it is called a tetrasomy. Tetrasomy is a condition of 2n plus 2. Monosomy is a condition, that is 2n minus 1. Nullisomy 2n minus 2. Trisomy 2n plus 1. Question number 33. V-shaped chromosomes are produced by, they are produced by metacentric chromosomes. Here we can see metacentric produces V-shaped chromosomes, submetacentric produce an L-shaped chromosome, acrocentric a T-shaped chromosome, and telocentric is an I-shaped chromosome. The structural and functional unit of chromosome is, it is a chromatid. A chromatid is two identical halves of a chromosome which are joined at a constriction region that is known as centromere. Question number 35. Isogenic lines are produced by, they are produced by back crosses. Question number 36. Organelles for protein synthesis is, it is called ribosome, which synthesizes the proteins. The process of mRNA production is called, it is known as transcription. The processing of hnRNA or heterogeneous RNA to mature RNA is called, it is known as splicing. The spliceosome enzymes are used in case of introns, whereas the mature RNA produces. The product of mass selection is genetically, the correct answer is homozygous heterogeneous population. Here the individuals will be homozygous, but as a population they will show variation. We can see here different types of populations. Where homogeneous are present, the individuals are also the same genotypes. No variation is seen. In case of heterozygous and homogeneous, the individuals, they are heterozygous, but as a population, they all are the same. In case of homozygous and heterogeneous, the product of mass selection, we can see there are individuals with capital T, small t, very rare. That means the population is heterogeneous and the individuals are homozygous. In case of heterozygous and heterogeneous, the population will be heterozygous. Also the individuals will carry two alleles. Question number 40. Recurrent selection is most commonly done in, it is done in cross-pollinated crops. Question number 41. Biparental mating was invented by, it was invented by Comstock and Robinson. Question number 42. Process reducing the frequency of crossing over is, it is done by the process of inversion. What happens in inversion is the chromosome segments breaks and it reattaches within the same chromosome but in a reverse orientation. Question number 43. Appearance of ancestral characters in progenies is known as, it is known as atavism. Atavism is the recurrence of a particular trait in an organism. Question number 44. F1 hybrids are always, they are always heterozygous. Question number 45. Which of the following forms of DNA has left-handed coiling? The correct answer is option number D, Z-DNA. We can see here the Z-DNA is left-handedly coiled, whereas the B-DNA is right-handedly coiled. Question number 46. In case of which of the following, complementary base pairing by hydrogen bond formation plays a critical role? The correct answer is option number D, DNA replication as well as transcription. It is very important that the complementary base, they are paired by hydrogen bonds. Question number 47. Transposons can be used for which of the following? They are the jumping genes which can be used for preparation of linkage maps, induction of mutation, isolation of genes. The correct answer is option D, all of the above. Question number 48. C value represents DNA content of a nucleus, that is in which of the following stages? The correct answer is option D, meiotic telophase 2. In meiotic telophase 2, we see that the amount of nuclear DNA in unreplicated gametic nucleus. Question number 49. Transposable elements generate which of the following changes in host DNA? The transposable elements, they move from one locus to another, causing the changes of duplication, deletion, as well as inversion. So the correct answer is option D, all of the above. Question number 50. Which of the following nuclei have 2C DNA? 2C DNA, it is a mass of DNA which is present in a haploid chromosome set. So the nuclei having those, they are present in somatic cell telophase and meiotic telophase 1. Thus the correct answer is option D, both A and B. Question number 51. The presence of replication fork in E. coli's chromosome was demonstrated by which of the following? The correct answer is option D, Keynes. Question number 50. DNA polymerase 1 enzyme was discovered by which of the following? The correct answer is option C, Korenberg. This DNA polymerase 1 enzyme, it processes the RNA primers during the lagging strand synthesis and repairs the DNA. Meselson and Stahl presented experimental evidence for the semi-conservative replication of DNA based on the study of, they studied E. coli, that is the Escherichia coli, which helped them to discover the semi-conservative nature of the DNA replication. Which of the following is expected to show least variation among its members? So they are the duplicate genes which show the least variation. Question number 55. The repeating unit of the rDNA of eukaryotes has which of the following? So it has 5' leader, 18S rRNA gene, 28S rRNA gene. Thus the correct answer is option number D, all of the above. Question number 56. Which of the following is generally accepted as a synonym for gene? The correct answer is option A, cistron. Cistron is a coding sequence of DNA which encodes polypeptides. Which of the following is represented by one nucleotide pair? So the correct answer is option number C, recon, which is known as the unit of recombination. The smallest unit of a gene capable of undergoing mutation is called, it is known as a muton. Lac operon is, it is an inducible system. It uses lactose as some source of energy and gets activated when the glucose levels are low but the lactose is present. Question number 60. Match the following concepts given by their ideal scientists. The crop idiotype concept was given by Donald. Gene for gene concept was given by H. Floor. The resistance concept was given by Harrington. The pedigree modified concept, that was given by van der Planck. And the double cross in maize, it was given by Jones. So that was it. Thank you. Hello everyone. Welcome again to the series of solved previous year's ICR-PG examination, Plant Science, where we will discuss the questions specifically on genetics and plant breeding. Today we will discuss the question paper of 2016. So let's get started.